Skeletons, as you know, are rare. Crania, as you've heard, are rare. Teeth are very common in the fossil record relative to other elements because they resist decay and destruction so well. There's good news and bad news in that. The good news is there are lots of teeth from lots of individuals, not just one female. The bad news is it takes an amazing, an extraordinary amount of work to squeeze biological information out of these hard tissues. And there is nobody more qualified to do that than our next speaker, Dr. Gen Suo from the University of Tokyo, who will be talking about the teeth. But there's more. He was also pivotal, not only to the reconstruction of the cranium, but also the analysis of the rest of the skeleton. So when Owen Lovejoy, scheduled to speak after Dr. Suo, told us on Monday that he was unable to make it to San Diego, I contacted Gen in Tokyo and asked him whether on his flight across the Pacific he might be able to do more than just his talk, but also Dr. Lovejoy's talk, to effectively morph into Lovejoy and talk about the postcranial skeleton. Nobody more qualified than Gen Suo to do that. So you'll see him morph in about 20 minutes into Owen Lovejoy and talk about the rest of the skeleton. Welcome to San Diego, Dr. Gen Suo.
Okay, thank you very much. I will focus the talk on Ardipithecus ramidus, which has somewhat considerably more available than the other earlier taxa. You have heard already about some of the similarities and differences about, for example, the Cadaba teeth and ramidus. However, taken in a broad perspective, compared to, for example, the African modern apes or Australopithecus, we think that we see broadly similar morphology and evolutionary grade. So with the somewhat more wealth of information available for ramidus, much of my talk will be focused on ramidus. Another focus, there are many things we could discuss, and I will go through some of them after the slide. A considerable amount of time I would like to spend on the phylogenetic position of ramidus. And this is because in our series of science papers, for example, the dentition, we focus more on the paleobiological aspects, and we did not talk too much about how we look at the teeth in order to infer the phylogenetic positions. But after our publications, we learned that there were some amount of skepticism about the position of these hominids here after the split with chimpanzees. And there were suggestions that it might be some kind of Miocene ape continuing through time, so on and so forth. So I would like to take this opportunity to add on and clarify some of those aspects. So what can we tell from teeth? Well, for example, social behavior. Behavior, they do not fossilize itself, except for in very rare instances like footprints. So it is very difficult to infer a lot about especially social behavior. However, we have already heard from Dr. Haile Selassie about the CP3 complex. Now let me explain a little bit more. The CP3 complex involves the upper and lower canines which interlock. The upper canine is sharpened against the lower premolar, which is often very elongated, so it actually serves to hone the upper canine all through life. Now this CP3 complex is present in all advanced primates, monkeys and apes, and the development of the complex varies according to their behavior and social pattern. Most notably, there are lots of male and female dimorphisms. Males who have to compete for access to females for mating will tend to have very developed canines. A very extreme example is found in mandrills, where there's a very insightful study published fairly recently that in mandrills, only males who are young and in their prime, for 3 or 4 years, they are able to sire actually offspring. And so once the canine wears down even a little bit, they start to lose their advantage in the competition for mates. Now that is probably a very extreme example. Mandrills are the most dimorphic in terms of body size as well as canine. However, this shows that the CP3 complex is actually very informative about competition levels, aggression levels, so on. And so Ardipithecus ramidus, there are enough canines to tell that, for example, this set of teeth is an example of male. And basically, Ardipithecus back teeth are pretty much about the size of humans, maybe a little bit larger, but not too much. The canines are about 30% larger at the base, and it is projecting more than humans. It is about the size of female apes, female chimpanzee, for example. And however, it does not hone anymore. There are worn examples which clearly show that the weaponry has already ceased to exist. So that tells us something about Ardipithecus and other primates as well. We can say a host of things about dietary adaptation and feeding ecology. For example, the frugivorous chimpanzees, they have inside, this is Ardipithecus, the same individual. Look, the upper tooth row and the lower tooth rows seen from the occlusal surface. The incisors of, for example, of frugivorous orangutans and chimpanzees are maybe about 25% larger. And so again, Ardipithecus seems to be a have a generalized dentition, not too specialized for certain diets. The cusp patterns are pretty conservative. It's not spiky like leaf eater or insectivorous primates. And it is a generalized frugivore, omnivore kind of morphology. And we look at a host of many things, not only the shape and size, but also internal structure, wear patterns, microscopic wear, enamel isotopic composition, and all these avenues of investigation suggest that Ardipithecus was not a heavy chewer, was not adapted to savanna kind of abrasive and hard foodstuffs like Australopithecus. So we think Ardipithecus teeth are exhibit a adaptation that is distinct from Australopithecus. Growth and development, we can look at the timing of eruption to infer certain aspects, and also teeth preserves the growth incremental markings as they grow, both the roots and the crowns. And that enables us to investigate things like environmental stress. I won't go into details at this time. And finally, phylogenetic inferences is what I would like to focus for the remainder of the talk. We look at external morphology, internal structure, and also the key word to how to infer phylogeny from these fossils is variation, how to interpret variation. And magnitude and pattern of variation is of prime importance. Also, we can trace these fossil lineages through time, which is how we test actual phylogenetic hypotheses. Variation, how to interpret it, that is where us specialists would differ from person to person, and why we would differ in our conclusions or interpretations. We spend a lot of time trying to input patterns of variation into our software up here, and before we output our conclusions. This, for example, are all the same tooth position of one Australopithecus species. I won't say which species because it doesn't matter for this purpose of discussion. For example, if you have a tooth row, if we make casts, good quality casts, we cut out just this position or just the second molar or just the third molar. And then we know that all of these are all coming from the same jaw part. All of these come from the second molar, all of these come from the third molar. They're all the same species. Now, how do you tell difference between M1, M2, and M3? And sometimes it's easier, sometimes it's more difficult. Different species have slightly different trends. And we have to have all that under control because we do not want to overread the fossil evidence. If you go for a different Australopithecus species, again, we can cut out just the M1. And so, for example, fossils do not come out with labels. If we mix these, how can we separate them? And we can to an extent, and we can't to an extent. And that is the balance that we have to always be worried about. For example, if you have a tooth row, these are all orangutan tooth rows, lower tooth rows. This individual has a smaller M1, bigger M2, and even bigger M3. This is a very small tooth individual of the same orangutan species. M1 and M2 is about similar size, M3 is much smaller. Now, if these came out from the ground in isolated, how would you tell apart? Or if it came out of the ground in tooth rows, if you found just this, you might say, oh, this species is characterized by big M3s. But if you have a series of teeth, then you know that it doesn't characterize the species itself. So you have to be very careful, and it's always an iterative process where you make a hypothesis saying, we think these features characterize the species, you find more, and then you refine it or you confirm it, so on and so forth. At the end of the day, there will be lots of overlap of variation in these features. And so it's, when you're working with limited sample sizes, it's a matter of how insightful you can be as to what might be the range of variation if you find more fossils. And you're always testing yourself and confirming and modifying. Now, with this confounding factor of variation, it's confounding, but it's also enlightening. So how do we deal with trying to find the correct phylogeny? One thing, there's no set protocol to this. Everybody has their own slightly different methodologies and inclinations. With the work with Hominids and others, at the end of the day, we are starting to rely more on, for example, extreme, distinct, and or rare morphologies. For example, a real example that we are applying, gorilla teeth have very high and pointy cusps because for a large-bodied ape, it has very unusual teeth that are adapted for shredding fibrous material or folding fibrous material into manageable swallowable chunks of food. So if you want to identify a gorilla ancestor, the place to look for is a morphological element, or element that has morphologies that have extreme morphology that would be evolving rapidly at some point of its history. Then you have a chance of being able to identify correctly an early member of that lineage. And so we are focusing into some of the fossils using those kind of approaches. With hominids, one place to look for is a CP3 because we know from an ape ancestry it turned into a female-like ape condition first and then progressively into a very small, underdeveloped complex like we see in modern humans. So for hominids, the canine complex is always very useful. Okay, so why do we think that Hominids is on the Australopithecus side of the pan split for sure? Relative to body size, male Ramidus canines are smaller than in any old world monkey or ape. Sexual dimorphism is weak or very little in shape. And this, again, is very unique. Functional honing is absent, as Dr. Haile Selassie has already introduced, so on and so forth. So one of the reasons we think Ramidus is hominid for sure is its CP3 complex. Another way of approaching teeth and phylogeny is you test for homology of the structure by structural analysis. If you examine the proposed similarities in more detail, if it does not have a common heritage, you would often find differences in the details of that feature. For example, enamel thickness. Ramidus enamel is thinner than Australopithecus. This is a 3D mapping of enamel thickness where we take micro CT scans of teeth in about 30 micron intervals, and then we separate the internal dentine surface from the enamel, and we make a enamel thickness map all along the crown. The red is thicker enamel, so you can see that the Ardipithecus ramidus enamel is much thinner than Australopithecus. We published about that without this comprehensive data in the 1990s. And then there were suggestions that maybe Ramidus thin enamel is because it shares it with chimpanzees, maybe it is an ape. So with a newer analysis, we did considerable more work and investigated this in more detail. And chimpanzees, it turns out, has thinner enamel than Ramidus, but also a very different pattern. It has a very wide occlusal basin with thin enamel, which effectively forms a very broad basin, which is suitable in crushing fruits. And also it keeps at the rim enough topography of the teeth, so it is also useful in shredding leaves. So we think that the chimpanzee condition is different from the Ramidus condition, which has thinner enamel, but in pattern is more similar to Australopithecus. Okay, the final test for phylogenetic hypothesis would be continuity through time. And for this, you need a sample to assess the variation and how the variation relates to different fossil assemblages. And for example, the canine and lower P3 morphologies of Ramidus are derived towards the direction of Australopithecus and phenetic details. And there's we think we can see a morphological continuity from around 6 million years to the Australopithecus age, around 3 to 4 million years. So we have already learned about the primitive Kadaba canine, which is basically at the female ape level of morphology. The Ramidus one canines get a little bit shorter. The basal size is about the same, but it still had very rugged lingual topography, reminiscent of the more primitive condition. When you get to 4 million year Anamensis, sometimes you get similar ones, other times you get more flatter spatulate canines, and in Afarensis it grades into even more spatulate and rounded corners, and so on and so forth. So we think we can trace the evolution through time, which also confirms or corroborates the phylogenetic hypothesis based on shared and not shared features. The same with the lower premolar. These are 3 examples of the Ramidus condition. This one is very asymmetric, and I don't have chimpanzees on the slide, but if we have chimpanzees, some will look very similar to that condition. This one is, from occlusal view, is relatively similar to that one, which is Afarensis. However, if you look at from the side, it is much more taller and more primitive in that regard. This one is very similar to Anamensis from occlusal view. Again, if you look at it from the side, it is a little bit taller, showing that it is not Anamensis. However, we can see that there is enough variation and it grades into the Australopithecus condition. So at all the elements, we can see this kind of transition from Ramidus to Australopithecus. And this is upper molars, again, Kadaba and Ramidus. If we mix them, this is an example where it would be very difficult to separate them. With Australopithecus and Ramidus, sometimes we will be able to separate them. If we have samples, we can characterize them pretty well, we think. Australopithecus has thicker enamel. It gets increasingly thicker from Anamensis to Afarensis. However, if you have a single molar and just mix it, and if I were to told, okay, what do you think about this? Well, I would not bet my life on it at all. Okay, lower M3 is the same kind of thing. So to summarize, we think we see a transition from Ramidus to Australopithecus, at least in terms of the teeth. With the lower limbs, the foot, we have that whopping grasping foot. The hip, we have a mosaic of features, hominid-like bipedal features, more primitive climbing features, as we have already heard. Some people think the 4.4 million Ramidus to 4.2 million Australopithecus transition would be too dramatic, given these body parts. The situation seems to be that the lower limb features, there may be a big change. Upper limbs, probably more subtle changes. And the dentition and parts of the cranium, the variation probably tends to grade into from Ramidus to Australopithecus. So we think that it still is not impossible that a 4.4 to 4.2 million transition may or may not have taken place. We cannot say that for sure, but we think that Ardipithecus ramidus, the condition as a species, is a very good potential ancestral condition for the Australopithecus lineage. It could have split earlier than the Aramis ramidus sample and then became Australopithecus. So to answer the question of what phylogenetic position Ramidus would have taken place, we think we can securely say it is after the pan split onto the hominid side. And this is a very basic question, but a question that we would like to have it cleared, because dependent on this position, the interpretation of everything else will change.